Hello students, welcome to 12th lecture of solid state. In this lecture we will discuss about calculation involving unit cell dimensions. So as we know, we already discussed about some of the dimensions of unit cell, and that was age of unit cells and angles between them, A, B, C and alpha, beta, gamma. In this unit we will discuss about some more dimensions of unit cell like density, number of atoms per unit cell, atoms, molecular or atomic mass of atoms or substituent particles of given unit cell. So let us discuss it step by step. So first of all, as mentioned in the textbook, from the unit cell dimensions it is possible to calculate the volume of unit cell. That means if you know the age length of unit cell, then as we know, unit cell is a cubic shape, so a cube will give us volume of given unit cell. Now if you know the density, we can calculate mass of atom in unit cell, and from the calculation of mass we can find molecular mass or atomic mass of given substituent particles. So we can find this by deriving equation, and it can be done by taking relationships between them. In this case we can find exact value of Avogadro number as well. We already know Avogadro number is a fixed number, that means it is constant, it do not change, but in certain examples intentionally different type of values will be mentioned. So if you calculate using equation which we are going to derive, then you may find different type of Avogadro number. So if Avogadro number is asked in question, you have to calculate using given values. Do not select direct answer. Okay, although we know the value of Avogadro number, but in question maybe different value will be obtained. So now let us discuss further. So suppose age length of unit cell of a cubic crystal determined by X-ray diffraction. Now what is X-ray diffraction? Let us discuss it shortly. So to observe something, normally we impact light on it. Suppose this is our particle size and this is wavelength of our visible light. So as we can see, wavelength is shorter than given particle. So in this case, wave or light will interact with given particle. Okay, and now you can see this particle. But if situation is opposite, size of particle is comparatively small than our given wavelength, then this wave normally do not interact with smaller sized particle. That means no change in wave or light will be observed, so you cannot detect that particle. So to overcome this situation, as we know unit cells are very tiny, we cannot see them normally, and the wavelength of visible lights are far more bigger than the size of particles, constituent particles and unit cell made by them. So that's why we need wavelengths short enough which can interact with smaller size particles, and that's why we will use X-rays. Wavelength of X-ray and size of particles are almost near to each other. That's why X-rays will interact with it. So we will use these type of smaller wavelength containing light radiation which can interact with small size particles. Okay, so using this method we can find age length. So we can find, using this method we can find age length, and if you can find age length, then we can derive some more information regarding to unit cell. Okay, so let us discuss it. So first of all, equation of density. So we know that density is equal to mass upon volume. So to find the density of unit cell, we are doing this for unit cell. We are finding unit cells dimension and calculations. So density of unit cell is equal to mass of unit cell divided by volume of unit cell. Now how can we find mass and volume of unit cell? So let us discuss it. So first of all, let us discuss about mass of unit cell. So mass of unit cell is usually obtained by constituent particles of unit cell. So constituent particles of unit cell have their own mass. Let us assume they are atoms. So mass of one atom multiply by number of atoms in unit cell, in given unit cell. So let us assume if we have primitive unit cell, then primitive unit cell contains only one atom. In FCC unit cell we have 4 atom, and in BCC unit cell we have 2 atoms per unit cell. Okay, so for primitive unit cell we have to use 1 as the number of atoms. For FCC we will use 4, and for BCC we will use 2. Okay, so that means mass of unit cell is equal to number of atom, let us call it Z, and mass of given atom, let us call it small m. Okay, so mass of unit cell is equal to Z into m, where Z represents number of atoms per unit cell. For primitive it is 1, for face centered it is 4, and for BCC it is 2, multiply by mass of single atom. Then next is volume of unit cell. Okay, density is equal to mass into volume. So volume is of unit cell is equal to A cube, where A is age length of unit cell. Okay, so by practically applying X-rays on substance we can find A, that means age length or distance between two nearby atoms. Then we can cube it, so you will get volume of unit cell. By placing this value in given equation we will get density of unit cell is equal to mass. Now mass of unit cell is equal to, as we discussed, Z into m, where Z is number of atoms per unit cell and small m is mass of single atom. So it will be Z into small m divided by volume of unit cell, which will be A cube. Now how can you find mass of single atom? So let us discuss about it. So let us use a practical example. Now we know that atomic number and atomic weight or mass represents total mass of 1 mole atom, and 1 mole is equal to 6.022 into 10 raised to 23. So as a simplification, let us consider a simple example of hydrogen. Now hydrogen atom, if you have 1 mole of them, that means if you have 6.022 into 10 raised to 23 atoms of hydrogen, then mass of this large number is equal to 1 gram for hydrogen. Okay, so if we want single mass of single atom, then you have to divide total mass, which is 1 gram, divided by total number, which is 6.022 into 10 raised to 23. Okay, more simple example, suppose you have a bag full of a certain fruit, let us say mango. We have a bag full of mangoes, and total mass of that bag is 10 kg, and number of mangoes in it, let us say we have 50 of them. Okay, so 50 mangoes and total 10 kg mass. So mass of single mango, it will be total mass divided by number of mangoes, which is 50. Okay, so 10 divided by 50, it is equal to 0.2 kgs, that means 200 grams per mango. Okay, so same thing can be done here. If you want to find mass of single atom, you need atomic mass divided by Avogadro number. Okay, that means atomic mass is mass of 1 mole of atom. So total mass of 1 mole divided by quantity or number in 1 mole. Using this equation, you can find mass of single atom. That means mass of single atom is equal to atomic mass capital M divided by Na, Avogadro number. Now let us put this value of small m into our equation. So our equation was D is equal to Z into M upon A cube. Okay. So let us replace value of m here, because m is equal to capital M, that means atomic mass, divided by Avogadro number. So let us put this value in equation. So our equation will be Z into capital M divided by A cube Na. Okay. So this is our final derived equation, where we have 5 different dimensions of unit cell: density of unit cell, number of atoms in unit cell, atomic mass of substituent particles, volume of unit cell, and Avogadro number. We have total 5 different values here. Okay, so this is the derivation of given equation. If you have any confusion regarding to this derivation, you can ask me. And now let us move on example. 3 examples are mentioned, but 2 of them are comparatively simple. So we will discuss about complicated example only. So first example is complicated, so let us discuss about it first. So example 1.3, an element has a body centered cubic structure. That means it has BCC structure. So number of atom, that means Z is equal to 2. Structure with the cell age of 288 pm. That means A is equal to 288 pm. The density of element is 7.2. So D is equal to 7.2, but unit is gram per centimeter cube. And how many atoms are present in total? 208 grams of the element. Okay, so these are the given values, and we have to find total number of atoms in 208 gram of element. So here first thing, value of density is mentioned in the reference of centimeter, while age length is mentioned in term of pm. So you have to convert pm into cm first. So first of all, we have age length of single unit cell, that means A, which is 288 pm, but as mentioned, density is mentioned in centimeters, so we have to convert pm into cm first. So 288 pm is equal to 288 into 10 raised to minus 10 cm, or 2.88 into 10 raised to minus 8 cm. Okay, so this will be age length in term of centimeters. And now we have to find the volume, that means A cube. So A cube is equal to, in term of centimeter, 2.39 into 10 raised to 23 centimeter cube. So this is the volume of single unit cell. Okay, once unit cell has mass, volume is equal to 2.39 into 10 raised to minus 3 23 centimeters cube. Now how can we find volume of 208 grams? That means total volume. So as we know, density is equal to mass upon volume. Now in this case, we have density of substance and mass of substance. That means you can find volume. So our equation will be volume is equal to mass upon density. Now mass of substance 208 grams and density of substance 7.2 gram per centimeter cube. So from this calculation, that means 208 divided by 7.2, you will get 28.88 cm cube of volume. So total 208 gram of substance has 28.88 centimeter cube of volume. That means in 28.88 centimeter cube, we have volume of all the unit cells present in compound. So how can we find number of unit cell from it? So first of all, let us discuss it with a simple example. Suppose we have let us say 100-liter tank. Okay, you have 100-liter tank and you have to fill it with 100 ml bottles. Okay, we have to fill 100-liter tank with 100 ml or milli bottles. So first of all, we have to convert them into same unit. So let us say 100 ml is equal to 0.1 liters. So you have to fill 100 liters tank with 0.1 liters bottles. So how can we find the number of bottles required? So total divided by 1, that means volume of total bottles required divided by volume of single bottle. So it is equal to 1000. We need 1000 0.1 liter bottles to fill up 100 liter tank. Okay, so same calculation can be done with unit cells. You have to do is total volume divided by volume of single, then you will get number of given substance particles or unit cells. So let us do it. So as discussed earlier, we have to do all you have to do is total volume of 208 gram of substance divided by volume of single unit cell, which is 2.39 into 10 raised to minus 23. So it is equal to number of unit cell in 208 gram of substance, and it will be 12.08 into 10 raised to 23. That means we have 12.08 into 10 raised to 23 unit cells in 208 grams of substance. Okay, so now last step, you have to calculate number of particles or constituent particles or atoms. So as mentioned in conditions, we have BCC unit cell, and as we know, in BCC unit cell, one unit cell contains two constituent particles, and here our particles are atoms. So number of unit cell multiplied by 2 will give us number of atoms. So number of unit cell here is 12.08 multiplied by 10 raised to 23 multiplied by 2 is equal to 24.16 into 10 raised to 23. So this is the number of atoms. Total in 208 grams of substance we have 24.16 into 10 raised to 23 number of atoms. Now remaining two examples are quite easy, so let us discuss about them quickly. So as mentioned, X-ray diffraction study shows that copper crystallizes in FCC unit cell. That means it will have 4 atoms per unit cell. Okay, so age length, that means A is equal to 3.608 into 10 raised to minus 8 centimeters. So it is already in centimeters. In a separate experiment, the copper is determined to have density of 8.92 gram per centimeter cube. Calculate the atomic mass of the copper. So all you have to do is, our initial equation was D is equal to Z into M divided by A cube Na. Okay, so our target is to find atomic mass, that means capital M here. So our derived equation will be M is equal to density multiplied by A cube multiplied by Na divided by Z. Okay, so derived equation will be M is equal to D into Na into A cube divided by Z, and all of the values are mentioned here. Density, it is already mentioned, 8.92. Na, we already know, 6.022 into 10 raised to 23. It is fixed value. A cube, from the value of A you can find A cube and Z. So as mentioned, FCC unit cell has 4 atoms per unit cell. So by putting the values, you can find value of M, that means atomic mass, which is 63.1 for given example. So comparatively easy calculation. So now next, silver forms CCP lattice. Now CCP, that means it will have FCC unit cell. So again Z is equal to 4 for given example. So X-ray studies of crystal shows that the age length of unit cell A is equal to 408.6 pm. So you have to convert pm into cm. So calculate the density of silver atomic mass, that means capital M is equal to 107.9. So first of all, as mentioned here, because of CCP, Z is equal to 4, molar mass 107.9 into 10 raised to minus 3 kg. You can use this value given here, mentioned here. Okay, no need to change it. In final equation, same type of value will be obtained. Okay, so one extra step is mentioned here. No need to convert molar mass into kg if it is not required or asked. Okay, so next, age length. So it is mentioned in pm, so that's why we have to convert it into cm. Here it is converted into meters, so again no need to do it if it is not asked. So our equation, density is equal to z into m upon a cube na. So our target is to find density, that's why same equation will be used. So all of the values are put here, and finally as mentioned here you will get 10.5 into 10 raised to 3 kg per meter cube. So as you can see, unit is kg per meter cube. So now we can convert this into gram per centimeter cube. It will be 10.5. Okay, so first in this calculation they converted all the values in kgs and meters, then in final equation they again convert them into gram and centimeter term. Okay, so no need to overcomplicate this. You can directly calculate using this value, gram per mole, it is already mentioned, and you can convert 408.6 pm into 408.6 into 10 raised to minus 10 cm. You can directly use this value in calculation. Okay, so instead of using kg per mole, you can use gram per mole unit and divide by this value. That means instead of using meter, we can use centimeter unit for age length. Okay, our next topic is imperfection in solids. So we will not discuss it in detail in this lecture, we will do it in next lecture. First let us get some basic description for imperfections in solid. So as mentioned in textbook description, so let us discuss it. Although the crystalline solids have short range as well as long range order in the arrangement of their constituent particle, yet crystals are not perfect. Okay, so you have to keep in mind, crystals are not perfect. They have some defects or faults in them. Usually if you have solid crystal, then it is not a continuous single crystalline structure. It is actually made up of some small sized crystals. Okay, very small and tiny crystals cluster or aggregate together, and then they form large sized crystals. Okay, so actually larger sized crystal is a aggregation or a bunch of small sized crystals. So these small crystals also have defect in them. As for example, suppose this is our small single crystal. So one more second crystal will combine with this like this, then third, fourth, fifth. These type of random small sized crystals will attach with each other, and eventually you will get a larger sized crystal which you can see. Okay, so that means all you can see is a single large size crystal is actually cluster or combination of small sized crystal. As for example, suppose you have a small sized NaCl crystal in your hand, you are holding it, suppose, so it will appear as a single crystal, but actually it is cluster of multiple small sized crystals. And if we zoom in this small sized crystal, then it will have some defects in it, and defects have two different types, point defect and line defect. So what is point and line defects? So suppose, just assume constituent particles in this crystal are arranged like this. Okay, that means let us assume it is two dimensional arrangement as mentioned in 2D close packing structures. Okay, so these type of particles are arranged in crystal, and if some of the particles are missing or some extra particles are present, as for example, suppose this particle is missing from its location. Okay, that means defect is observed at a single point. Or suppose this particle is present at unwanted location. That means it is not location of this particle, but it still occupies. So this is called defect at certain point. So these type of defects, that means if particle is missing or extra particle is present, so these type of single particle defects are called point defects, defect observed at certain point. But if defect is observed in entire row of particles, that means suppose these are the rows of particles. Okay, suppose if extra particle raw is present here, that means this should be empty. Okay, but one extra particle raw occupies this space, then it is called line defect. That means if entire line has a defect, then it is called line defect, and if certain points have defects, then it is called point defect. So in this unit we will discuss about point defects only.